Thanks for having me on, Rochelle.
So briefly explain to viewers just how does the drug remdesivir work?
So remdesivir basically blocks the virus from making virus children. So it results in chain termination by faking the virus into thinking it's a building block when actually it tries to use it to build more of itself and it falls flat.
And clearly there's a lot of different approaches being tried. We know that there are other trials ongoing currently, including cell therapy. What are the main differences in the most promising options available?
So I would think about the Olympics of treatment right now, and remdesivir is probably a silver medalist. And I would think about the others, including convalescent plasma, maybe stem cells, as potentially being bronze medalists. They're not antivirus drugs. They're really attacking they're modulating the immune effects. So they're not really attacking the virus itself. But at the end of the day, we probably need a little bit of everything to have the complete picture.
So then what sort of dangers are there, if any, with all these trials?
I think if you try to mess around with the immune system too much, as some of the sites have been using, I think out of no other options, using some of the immune drugs outside of a clinical trial, it could be a little bit dangerous. When we tried to use immune drugs in sepsis before, some patients actually got worse. So I would say that if people are using it, to try and use it within a trial.
Now, as we know, cost is likely going to play a huge role in who gets the vaccine first. So once a vaccine is widely available, which areas or groups do you think will likely be first in line to qualify for it?
That's a very difficult question, but I think a lot of ethicists are thinking about a principle of ethics called the multiplier effect. So that means if you treat certain populations, you can have benefits beyond the populations you treat. So one population people have been thinking about are healthcare workers. So if we vaccinate them and they're protected, they can then treat many more people beyond themselves. So I think really that healthcare workers should be one of the biggest targets, and then some of the groups that do poorly with the disease. So elderly folks, folks with comorbidities, maybe another level that we can think about.
Yeah, we certainly won't get too far if our frontline medical workers aren't there to help the rest of us. Now, we know that manufacturers are going to have to produce the vaccine in very large quantities. Are there any companies with that kind of capacity, or can we expect more collaborations?
Yeah, so I really think this is an area where you need to have collaboration. Of course, vaccine companies, people like Sanofi or Pfizer, that they're used to giving vaccines, they are probably working in collaboration. You've been seeing this happen already with a Pfizer-German collaboration. So someone in a smart lab will come with a vaccine candidate, but it's really up to big pharma to make that in huge numbers and to use a distribution network that can get to a lot of people quickly, particularly if you need a cold chain.
So I also want to talk about ventilators and PPE, personal protective equipment. We know that they're currently the top priorities for the healthcare sector in dealing with COVID-19. Once a vaccine exists, what else is likely to be needed to carry out the next steps?
So I think other things that people are looking at would include, again, one is a little bit old-fashioned, which is contact tracing. We've talked a little bit about that. And the other is enough testing. It seems we've been talking about testing over and over again, but we need more tests for the virus as well as tests for antibodies. And then I think we get a spectrum of what's going on in the community and whether or not, particularly with antibody testing, if we have enough herd immunity to know that the vaccine is actually working.
So then if remdesivir does happen to be successful, and it was successful in curing SARS and MERS, why do you think it was so difficult for researchers to really prepare for COVID-19?
Well, it was lucky that Gilead had some remdesivir in the freezer because, of course, it didn't work quite as well in Ebola. But it did have activity in SARS and MERS. So again, because it didn't have activity in everything, I think one has to really study it before you can really make any statements. Every virus is a little bit different, and just because it worked in another virus doesn't make me want to know for sure if it works in a completely new or novel virus.